Today we have a lineup of great speakers, and hopefully that would be an interesting discussion at the end as well on how we might be able to move forward with precision functional mapping for individualized neuromodulation. The first series of talks are going to be some core talks, and we have a kind of a list of speakers for the core talks. And the first opening talk would be my co-chair, Charlie Stagg from Oxford. Charlie, please.
Thank you, and thank you very much for the invitation to come along and do this. It's a real pleasure, and I'm very much looking forward to hearing the talks. I think by the fact that so many people are here, people probably are very bought into the idea that we need two individualized brain stimulation technologies, and that that's going to be a very important step in maximizing outcome. But Hamida asked me to sort of introduce the topic. So what I thought I'd do is really just kind of do a whistle stop through just a couple of examples of why this matters, right? I think hopefully people are here are on board with the idea that neuromodulation and brain stimulation can do significant things to the cortex, and that can be useful in a range of neurological and psychiatric situations. And this really is a classic example of theta burst stimulation. These are the results from the first study coming out of John Rothwell's lab a long time ago, and they were absolutely beautiful. Right, so if you did intermittent TBS, you got a nice increase of excitability. If you did continuous TBS, you got a decrease in excitability, and the sham was beautifully in the middle. But we all know that's not true. Okay, and we've, the more we do brain stimulation, the more we realize how incredibly variable these responses are. So next slide, please. So if we just look at for example, continuous theta burst, this is a subsequent study coming out of the same group, and you can see that yes, some people do indeed decrease their excitability in response to CTBS, but lots of people don't. And it's very difficult really to see how, given this variability in response, how we might ever find a brain stimulation technique that is going to be useful across a clinical population. And so what we really need to do then is look at what this variability is telling us. And I think quite often it's telling us something useful. Okay, so hidden within this variability is useful information. So next slide, please. So in our work, we've looked at change in inhibition in response to anidal TDCS. And a long time ago, we and subsequently others have shown that the amount to which you can decrease your inhibition in response to anidal TDCS predicts how much you can learn. Okay, so the variability from subject to subject in response is really important, and it tells us a lot about people. But next slide, please. If we look at the response of GABA to anidal TDCS, we see actually that about 50% of that variability is not due to interesting inter-subject variability, it's due to how much E-field actually reaches the part of the brain that we're recording from. So this is recent work in brain stimulation where, as you might imagine, the higher the E-field in the area we're recording, the greater the change. So it's going to be really important to deal with this sort of almost 50% variability to target our brain stimulation in a whole host of ways, spatially, temporally, frequency, all kinds of state dependency. We'll hear a whole host of things, but it's going to be very important to do that so that we can A, reduce that variability, and or B, understand what it's telling us about the underlying subjects. So that's my little spiel. I'm looking forward to listening to everybody who's going to tell us the answer. Thank you.
Thank you very much, Charlie, for opening the session and helping me to co-chair the discussion that we are going to have. Hopefully, we will be using the chat space to kind of ask questions and keep engaged in the discussions that we have, and we will have time at the end to discuss about the questions and things that we can do together. So I will have a kind of a short opening talk as well, discussing about what are the challenges and hopes that we have for individualized neuromodulation? What is the pipeline for being able to do individualized neuromodulation? I do not have any conflict of interest. When we talk about individualized neuromodulation pipeline, you'll have at least 5 major steps to take. The first step would be defining, okay, what is the target biomarker that we are interested to individualize our stimulation protocol upon that specific target. And we will kind of hear today from different speakers about different potential targets that could be kind of used for individualization. But we need to have a hypothesis, I mean, based on the literature, based on what people have done so far. And people kind of mainly use different neuroimaging modalities, but there are still many questions in terms of what should be the characteristics of target biomarker. And as you know, for example, when we talk about resting state fMRI as a target and connectivities that we can kind of extract from resting state fMRI data, there is already an ongoing discussion in terms of how much that data should be reliable. Do we need to collect resting state fMRI for something like 40 minutes to have a reliable data, or just a state kind of dependent short resting state fMRI would also be sufficient to be used for individualized targets. So, and this is the case for task-based fMRI as well. And people try to develop different specifics of fMRI tasks to make sure they will have reliable data. And the question is how much reliability is important or not important. So we'll discuss about those issues during the talks today. The next step is, okay, how we might be able to optimize the localization of that specific individualized target. And as you can imagine, there are different methods and kind of the ways that we can target that specific location. But beyond that, we will also have discussions on how to optimize the dose and how to optimize the amount of electrical current that we are delivering into different parts of the brain. And there are different discussions. Today, we will have speakers discussing about how coil orientation is going to be important, how the intensity should be kind of optimized in the individual level, the amount of electrical, the amount of kind of the power or energy that we are putting inside the brain. So there are lots of discussions about dose optimization. Do we need to kind of optimize the dose or not? And then there are ongoing discussions regarding the experimental design. When we kind of run an individualized neuromodulation pipeline, what should be the control condition? Is it just enough to have a sham condition as a control, or do we need an active control? And then when we talk about an active control, the active control should be just another site, or we are thinking about a non-optimized active condition, and how people can basically and you always have limited resources when you want to run a trial, and how many control conditions you need to consider in an individualized neuromodulation pipeline to make sure that individualization is the factor that is making the change. And then at the end, there are questions in terms of how we test the hypothesis that we develop at the first place, and what are the changes that we are expecting to see. For example, if we targeted a specific connectivity, are we going to see that connectivity is strengthening or weakening, or are we expecting to have some changes in activation inside the brain, and how that specific response biomarker is going to be validated? And are we expecting to have a response biomarker to be the same as target biomarker or not, and how they might kind of correspond to each other? So there are still discussions on that side. In my own database, I already have something like 33 real individualized TMS fMRI studies that people optimize the TMS protocol based on fMRI. But we know that there are still really limited amount of kind of actual published randomized clinical trials that people try to see how their kind of the individualized TMS protocol could make a change in terms of the final effect. So we'll discuss about some of those issues during the webinar, and hopefully at the end of the webinar we will have those discussions. And we are hopeful that within the kind of the international network of neuromodulation, we will be able to provide some opportunities for ongoing discussion on how to optimize the methodological rigor in that specific field. Those who are interested, we have a specific WhatsApp group, so you can just scan the code and join the WhatsApp group that we have. We are planning for some activities in OHBM. Those who are in the WhatsApp group, they will be informed about the things that we are planning for OHBM. We are putting together a study group in ISMRM. So those who are a part of ISMRM community, they can join us for those specific activities. We will have a panel in SOBP in April, I mean, just a few weeks from now, maybe 2 weeks from now. So those who are coming to SOBP will definitely be happy to continue discussions there. Now we are putting a panel with a study group, with a group of friends, colleagues in ACMP meeting. So we will try to kind of be active on those areas. And those who know about the activities that we are doing, you know that we had a specific webinar on dose response in brain stimulation studies in November, a series of webinars discussing about that in November. And now we are writing a white paper with those people who have been involved on that webinar and some others who have been interested. And I'm hopeful from this webinar, this specific webinar that we already have, those who are interested in a specific topic, we can have another white paper discussing about the challenges and hopes and the specific methodological issues regarding individualized neuromodulation. So those who are interested, you can just follow us on Twitter and on YouTube, so you can have the codes. I will provide the codes at the end of the webinar as well.